Alzheimer's disease presents the brain's immune system with two concurrent failures. The tissue suffers from chronic, toxic neuroinflammation, while simultaneously failing to mount the cellular response needed to clear amyloid plaques. The chronic inflammation represents a fire. It degrades synapses, accelerates neurodegeneration, and requires an immune intervention to put it out. The accumulating amyloid represents a biological debris field. Clearing it requires a highly active repair crew to surround the plaques, compact them, and break them down. Governing these responses is the regulatory T cell, marked by the transcription factor FOXP3. It is the immune system's master brake, a cell specialized to enforce tolerance and stand down immune activity once a threat has passed. This creates a direct theoretical tension. If Alzheimer's is a disease of excess inflammation, the regulatory T cell is a guardian that puts out the fire. If the disease is a failure to summon a repair crew, this same peacekeeper is a saboteur holding the workforce back. This tension fractured the field of neuroimmunology. Two prominent research camps ran the exact same experiment, depleting regulatory T cells in Alzheimer's mouse models, and reached opposite conclusions. One camp, led by Michael Schwartz at the Weizmann Institute, observed that depleting these cells cleared cerebral amyloid and improved cognition. They concluded the regulatory T cell stands in the way of healing and must be removed. The opposing camp, led by Guillaume Dorothy and others, observed that depleting the exact same cells accelerated cognitive decline. They concluded the regulatory T cell protects the brain. Both of these peer-reviewed results are real. Both are factually correct. Reconciling them requires tracing the regulatory T cell as a highly sensitive rheostat, governed by strict variables of timing, compartment, and disease stage. The thesis casting the regulatory T cell as a saboteur originates from a foundational 2015 experiment by the Schwartz Laboratory. The first critical variable in their data is the specific animal model they used, the 5X aphid mouse. This is an aggressive model where amyloid pathology accumulates overwhelmingly fast, rapidly outpacing the capacity of the brain's resident immune cells to respond. The second critical variable is the temporal structure of the intervention. They applied a transient, brief depletion of systemic regulatory T cells in the blood. They did not permanently remove them. This diagram illustrates the mechanism they discovered. Briefly lowering systemic immunosuppression acts to open the brain's border organ, the choroid plexus. Through this open gate, a wave of peripheral, monocyte-derived macrophages crosses into the central nervous system. Because the endogenous cells were overwhelmed, this sudden influx of an external myeloid workforce physically clusters around the dense amyloid plaques and efficiently clears them. This success produced a flagship therapeutic corollary. Researchers proposed using PD-1 checkpoint blockade, a tool common in oncology, to artificially break immune tolerance and unleash this reparative wave in human patients. In 2018, a multi-pharma consortium led by Lotte Mailloux attempted an industrial-scale replication of this PD-1 blockade. Across multiple models and laboratories, they found no macrophage infiltration and no change in amyloid pathology. A transient pulse of immune activation can indeed force a reparative wave in a rapidly overwhelmed brain model, but attempting to permanently break systemic immune tolerance as a monotherapy failed. The opposing argument, developed by the Dorothy and Bake laboratories, approaches the system under different constraints and casts the cell as a neuroprotective guardian. Their first contrasting variable is the animal model. They utilized APP-PS1 and 3-TAX-TGAD mice, where amyloid pathology accumulates much slower. This pace allows the brain's resident immune cells the time required to mount a meaningful reaction. Their second contrasting variable is the duration of the intervention. Rather than a brief pulse, they initiated a sustained chronic depletion of regulatory T cells lasting for months. The biological consequence of this chronic loss was severe. Sustained depletion ruined the brain's endogenous response, actively preventing resident microglia from effectively clustering around the plaques to contain them. These PET scans capture the clinical outcome. The removal of regulatory T cells caused a severe deterioration in cerebral glucose metabolism, providing physical proof of an accelerated cognitive decline. These cells provide direct trophic support to the brain parenchyma itself. Brain-resident regulatory T cells acquire a specialized tissue-adapted program that serves homeostasis and repair. These cells secrete a repair factor called amphiregulin. This molecule acts directly on astrocytes, actively suppressing the neurotoxic astrogliosis that otherwise worsens tissue injury. When the brain relies on its own resident workforce to handle a slow pathology, the sustained presence of these regulatory cells is mandatory. Without them, toxic inflammation runs unchecked, and tissue repair halts entirely. Combining the specific variables from both laboratories into a single matrix provides a unified biological model. A transient dip merely triggers a beneficial rebound response, allowing a brief influx of repair cells. But a chronic loss removes a permanent, necessary protection, leaving the brain vulnerable to its own inflammation. The compartment matters equally. Temporarily opening a peripheral gate at the choroid plexus is a distinct intervention from removing local, central support cells from the brain tissue itself. The regulatory T cell acts exactly like a rheostat. Its capacity to shelter the brain or starve it of repair inverts entirely based on where the knob is set and how fast it is turned. Applying this rheostat model to human patients requires shifting from controlled transgenic mice to cross-sectional clinical data to see how the knob is naturally set in the human disease. Initially, studies attempting to count the raw frequency of regulatory T cells in human blood yielded noisy results, with different cohorts showing increases, decreases, or no change at all. The decisive metric here is suppressive capacity, the actual efficiency with which each Treg restrains responder cells. Upon reaching the established dementia stage, this function definitively drops. In established Alzheimer's disease, the Peacekeeper functionally fails, leaving the brain exposed to chronic inflammation. Because this biological break is failing, the therapeutic strategy for humans must be to restore and amplify the cell's function, rather than depleting it further. The most viable pharmacological tool to achieve this amplification is low-dose interleukin-2. This therapy specifically exploits the high-affinity CD25 receptor, uniquely present on regulatory T cells. The IL-2 molecules latch onto these receptors, triggering rapid cellular expansion without activating the surrounding effector cells. By maintaining a low dose, the therapy targets the failing Peacekeeper while avoiding the risk of unleashing an anti-amyloid autoimmune meningoencephalitis. In 2025, a Phase 2A human trial led by Fari Dar tested this theory in patients with mild to moderate Alzheimer's disease. The trial demonstrated that low-dose interleukin-2 was safe to administer. It expanded functional regulatory T cells, shifted plasma inflammatory biomarkers favorably, and trended towards stabilizing cognitive decline. The Alzheimer's brain demands peace and repair simultaneously. The future of neuroimmunology relies on finding the therapeutic optimum of the regulatory T cell, giving the brain the intervention it needs, exactly when it needs it.